Urease activity test. This is another biochemical test for characterization or classification of microbes. Urea is a major organic waste of protein digestion, and it is excreted in our urine. There are microorganisms which have the ability to produce an enzyme called urease. This enzyme is a hydrolytic enzyme and attacks the carbon and nitrogen with liberation of ammonia. So urease is an enzyme which attacks on the carbon and nitrogen of the urea to liberate ammonia. The test is performed by growing the isolate or the microbe or the test organism on a broth called as urea broth, provided the medium should contain a pH indicator phenol red, pH 6.8. If the microorganism possesses this enzyme urease or have the capability to produce urease, as I have mentioned earlier, then ammonia will be produced, which will ultimately raise the pH of the medium or the broth, and therefore phenol red changes from yellow to a red or deep pink color. So this change in the pH changes the color of the indicator from yellow to a red or deep pink color. So this test confirms positive test. Requirements: 24 to 48 hours slant culture of microorganisms, inoculating loop and Bunsen burner. Procedure: Christensen's urea agar prepared using the following composition in 1000 ml: peptone 1 gram, dextrose 1 gram, sodium chloride 5 gram, potassium phosphate monobasic 2 gram, urea 20 gram, phenol red 0.012 grams, agar 15 to 20 gram. This is for 1000 ml and this is Christensen's urea agar. Now to prepare this urease base, dissolve the first 6 ingredients in 10 ml distilled water and filter sterilize. Remember, in the ingredients, the first 6 ingredients to be dissolved in 10 ml distilled water. Pour agar in another conical flask in 90 ml distilled water and autoclave. Cool for 50 to 55 degrees centigrade. Now the urease base to be added to the cooled agar solution and mix thoroughly and pour as slants.